Okay. Okay. So, okay, you can find the record, and please remember me to start the record every time, because sometimes I can forget it. Oh, medicinal chemistry is a chemistry-based discipline. So the starting point is chemistry. Also involving aspects of biological, medical, and pharmaceutical sciences. This means that you have to remember some topics from biochemistry, from physiology, because we will try to explain how drug acts, how drug is able to provide a specific therapeutical benefit. And so it is important to know the target. Hello, good morning. It is very important to know the structure of the target. And you know the targets we'll see later are proteins, receptors, and so on. So, however, medicinal chemistry, it is concerned with the invention, discovery, design. Design in Italian we say progettazione. Identification and preparation of biological active compounds. The study of their metabolism. The interpretation of their mode of action at the molecular level, and the construction of structure-activity relationships, SAR. This is an important point. Structure-activity. We will see during the course a lot of examples how we can modify the chemical structure of specific compounds in order to improve the potency, to improve the ability to interact with a specific target. For example, we can introduce different substituents on the aromatic ring in order to have different electronic effects, and we try to observe the effect with the target. This is an example. So the concept of structure-activity relationships is peculiar of medicinal chemistry. This is the definition of a IUPAC, IUPAC, you know. But in particular, for us, for you, what is medicinal chemistry? A simplified definition: design, progettazione for Italian students, and synthesis of biological active molecules to address unmet medical needs. For example, there is a strong research on Alzheimer's disease. You know, people become more older and older, and so we need new medicine, new drugs against this disease. There is a strong research against viruses, you know. Unmet medical needs. But today, there is a more modern definition, not only making new bioactive molecules, but also improving existing pharmaceuticals, old drugs, but new application. the medicinal chemists try to optimize the structure, the properties of molecules to improve their interaction with targets, as well as their metabolism to avoid side effects. Have you never read the paper inside the box of the drugs? There is a sheet very long with all side effects inside. And after reading, normally you don't want to take the drug because you can find a lot of side effects. Limited side effects. Medicinal chemistry is able to orchestrate a lot of different scientists. We need biochemists to know the structure of the targets, proteins, for example, gene. The structural biologists, clinical experts, pharmacologists, toxicologists. How to set up new experiments to evaluate the potency of a specific drug. Cell and microbiologists, computational analytical chemists. Computational chemistry is a very important branch of medicinal chemistry, because if you know the chemical structure, if you have a picture of a protein, it's possible to design new compounds by computational medicinal chemistry. The components of pharmaceutical chemistry. Organic chemistry, drug metabolism. We'll see that all compounds that we introduce in our body will be transformed by our enzymatic system. And so we have to know how specific compound, how drugs are transformed by our liver, for example. Quality control, it's important in the production phase. Pharmacology, the study of how chemicals affect the biological system. Drug discovery, of course, the discovery of new entities or new drugs. Pharmacokinetics, the distribution of the drug inside the body. If you take a drug, this drug is distributed in all organisms, from hair to feet, finger. And so you can identify this compound everywhere in our body. Pharmacodynamics is the phase related to the interaction of the drug with the target. Dynamics in Greek means force. So pharmacodynamics is the force of your drug, the ability to interact with the target and to activate a specific receptor and so on. What is a medicinal chemist and what does he do? You see in the picture, at the beginning, if you want to discover a new drug, you have to read a lot of paper about the different targets. You have to investigate the structure of your target in order to design a new chemical entity able to interact with this target and hopefully to activate this target. And so we put all together the different chemical compounds. We put all together in one pot, and then finally we have a drug. What's about our course? There is a definition. This is an Italian definition. We elaborated this definition 20 years ago. And here you can see the meaning of medicinal chemistry in university, and especially for the course of pharmacy. Medicinal chemistry studies products of natural products, of course, biotechnological and synthetic origin with biological activity, developing the design, synthesis, study of properties, mechanism of action at molecular level, chemical toxicological aspects, the use and the relationships between chemical structure and biological activity. We try to put together organic chemistry and biological activity. This is our bridge. This is a bridge. We have to put together these two different disciplines. And biological activity are the main classes of drugs. Moreover, the study will focus on the extractive and synthetic preparation of drugs, the analysis of substances with biological activity, and in particular of drugs and their metabolites. Okay. The most important point for you, examination modalities. During the course, I will try to explain you about 2024 chemical synthesis of drugs. And during the exam, I ask you two of these syntheses, one of your choice, and the second it's my choice. And so you have to demonstrate your ability to synthesize a drug, how a drug can be synthesized. After that, I ask you orally 3 topics, 3 chapters of the specific part of the course. For example, anti-inflammatory, anti-cancer drug agents, hypnotics. It's a very general question. It's a class. And so you have to organize your idea. You have to know the chemical structure of the most important compound of the class. Medicinal chemistry is like another language. It's like to study Chinese. And so you need a lot of exercise to write the chemical formulas. And during my university course, normally in my bedroom, I have a lot of paper on the wall with the different classes, and every evening I repeat every class. Because if you start to study this discipline, you believe to know a chapter, for example, penicillin, antibiotics, but the day after, you don't remember anything. So, when you read, okay, yes, this is one. The day after, nothing. Really. And so, it's my suggestion to prepare a summary of every chapter. Every evening, you have to stay at home. Okay, to repeat. Really, it's my suggestion. My personal suggestion. And at the end, when you are almost ready for the examination, I suggest to meet with your colleague together to ask the questions. It's very important. Just a moment. I wanted to share a brief movie. Where is here? Okay. Include the audio and computer. Yes.
So what is medicinal chemistry? The fancy term for it is it's the molecular bridge between biology and the clinical science of medicine. The simple layman's term for medicinal chemistry is it's the design and development of molecules that bind to a target protein with exquisite selectivity and huge potency. And the result of this complexation event is a biological response. Why is it important to be selective? Well, if you have a promiscuous drug that binds to every single protein inside the cell, you're going to affect a lot of vital biological processes. The result of that is toxicity. Why do we want it to be potent? So we only need to use a small amount to elicit that effect. If I have to dose a patient with a kilogram of drug, not only is it unfeasible to produce that amount of material, but if you're putting in that much material, you're going to have side effects because there's no way that it's not going to interact with other targets. So it has to be a combination of potency and selectivity.
So these are the two most important words, potency and selectivity. We need a potent compound, but selective. Otherwise, we have a lot of side effects. Side effects are due by the interaction of a compound with a lot of targets. This is the problem. High potency, but very selective. And normally we use small molecules. And so it's very difficult to increase the potency, to increase the chemical features, able to interact with a specific target, to avoid the side effects. What's about the goals of the course? At the end of the course, when you get your score of the exam, what kind of ability have you acquired? The course is aimed at studying the molecular mechanism of action of a drug, if possible, and in particular the types of bonds and steric factors involved in the drug molecular target interaction. It's important for you that when you see a molecule, a drug, a complex structure, you have to imagine what kind of bonds this compound is able to establish with a specific target. And this operation is very important also to work in a lab, to know the solubility, to know the chemical properties of the specific compound. The main classes of drugs that interact with targets in host cell and drugs that interact with the neurotransmitter receptor are considered. So we'll see a long series of different drugs. Of these drugs are studied the general synthesis, we'll see some examples, physicochemical properties, mechanism of action, structure-activity relationships, once again, this important concept, as well as the utilization of the chemical toxicological aspects. Okay. Propedeuticities, knowledge. Professor, I don't pass organic chemistry. Can I make your exam? No. No. No alphabet. You don't know any words. It is necessary to pass organic chemistry if you want to make my exam. It is necessary to know anatomy. When we speak about liver, you have to know the function of the liver, where is physiology, how the cell can be activated, general chemistry, biochemistry, what is an enzyme, what is a protein, which are the main amino acids in a specific protein. These are important concepts that you have to know. So it's important that you have this type of knowledge to follow the lessons and to understand the concept. Materials and texts. All slides are on the web. I try to record my lessons so you can listen again at home my lessons. Reference books are on the web. You can find also in the library here in the university if you want to see. You can buy this book, the Foyer book, also on Amazon. It's present. If you need to speak with me, you see my email, Skype, Teams. We can arrange an appointment, so it's no problem. I can explain you what you want. And very quickly, communication with Caterina. Grazie, Caterina, thanks. Just if we have some problems on the train line and so on. Okay. Yes?
Are we going to start every day at 10 or it was just today?
No, we start at 10 o'clock. So, yes. 3 hours are enough, really. Another language, after 3 hours, okay. My brain is completely over, but also your brain. 3 years ago, I started by teaching to the course of cosmetic sciences. After 3 hours, I go on the beach and I see the sea because really I was completely fused. Yes, at the beginning. Okay. Oh, this is the recommended book, Principle of Medicinal Chemistry. You can use this book for the 2 courses of medicinal chemistry, and so I suggest. It's just a little bit, there is a lot of pharmacology inside of this book. Few chemistry, a lot of pharmacology, but it's the reference book in the world. So, yes?
Do you also recommend that we should also study from the book more than the slide?
No. You can use the book as a reference if you want to know additional information. You see, if you want to study additional information. But the slides are enough. But you know, slides are prepared by me, and so the slides can reflect my personal point of view. The book, there are a lot of different authors, you see, very important scientists. Every chapter is developed by specific scientist, expert in that field, and so if you want to know more additional information, you can read the book. I took some picture from the book, so sometimes about metabolism and so on, transformation. It's very big, so 900 pages, I think, 1,000. So the course is organized in two parts, two main parts. The introduction part, where you can learn the alphabet of medicinal chemistry, the concepts of medicinal chemistry. And a second part, very specific, with different topics, with different drugs. And so sometimes during the second part, I can recall a lot of concepts from the first part. Normally, I don't ask anything about the first part. But if you don't know the first part, you cannot understand the second part. So it's obvious that you have to know the general concept. You see the different topics: molecular mechanism of action of a drug, pharmacokinetics, absorption, distribution, metabolism, phase 1 and phase 2 reactions, excretion, drug pollution. It's a urgent problem in our world, and we'll see. Drug receptor interaction, types of bonds involved, steric factors affecting drug activity, optical, geometric, conformational isomery, bioisostery, enzymatic inhibition, competitive and non-competitive inhibition, antimetabolites, enzyme inhibitors, analog of transition structures, receptors, intracellular receptors, and finally new drugs. Okay. These are the main chapter of the first part of the course. And now we start with the milestone of medicinal chemistry. You see at the beginning the publication of the first pharmacopoeia in the world, English, London Pharmacopoeia, 1600. Then the development of organic chemistry. An important German scientist, Justus von Liebig, German chemist and academic. 1860 First International Congress of Chemistry. And then the beginning of the new synthetic drugs. The first drugs were DET letter, anesthetic, acetyl salicylic acid, aspirin, you know, an hypnoid and a barbiturate, diethyl barbituric acid, a drug for sleep, for sleeping. And at the beginning of the 20th century, the birth of the modern chemotherapy. Chemotherapy is an important concept. I try to hit an exogenous source of disease, a virus, bacteria, fungi, and so on. So I have to prepare a specific bullets, a specific weapon, a smart weapon, just to hit the virus, the cancer cells, And Paul Ehrlich was the first scientist which hypothesized the possibility to hit these exogenous sources of disease. Some important achievements of medicinal chemistry. Discovery of sulfonamides. Sulfonamides were the first antibacterial drugs you see during the Second World War. Really, before that, nothing. So if you had an infection in that time, no solution, no therapy. 1942, synthesis of anti-malarials. You know, malaria is a disease from mosquitoes. The advent of penicillin G. American soldiers, they used this antibiotic, and they introduced this antibiotic in Italy during the Second World War. And they saved a lot of people. My mother was saved from penicillin. She had in that time a strong infection. And so Tumiati is here because my mother was saved by penicillin. Production of semi-synthetic steroid hormones. Discovery of isoniazid is the first anti-tubercular drug. Broad spectrum antibiotics. Chloramphenicol, we'll see this molecule. Oxytetracycline, we'll see. Ampicillin, cephaloridine. 1960, the discovery of the benzodiazepines. These compounds are very important. They are anxiolytic drugs. So they are used today instead of barbiturates, because barbiturates have a severe side effects. They are very famous. The commercial name is Tabor. I don't know if you see the box, perhaps at home. Your parents normally use these compounds when you are on the discotheque during the night, and so they want to sleep, and they take a pill. Discovery of nalidixic acid. It's another antibacterial synthesis, antibacterial drug. Fluoroquinolones, the same class. Imidazole antifungals, ketoconazole, okay, to fight the fungi infection in our body. And then introduction therapy of AZT is an important drug against IDS. Okay, I have another.
Packed inside every cell in your body is a set of genetic instructions, 3.2 billion base pairs long. Deciphering these directions would be a monumental task, but could offer unprecedented insight about the human body. In 1990, a consortium of 20 international research centers embarked on the world's largest biological collaboration to accomplish this mission. The Human Genome Project proposed to sequence the entire human genome over 15 years with $3 billion of public funds. Then, 7 years before its scheduled completion, a private company called Celera announced that they could accomplish the same goal in just 3 years and at a fraction of the cost. The two camps discussed a joint venture, but talks quickly fell apart as disagreements arose over legal and ethical issues of genetic property. And so the race began. Though both teams used the same technology to sequence the entire human genome, it was their strategies that made all the difference. Their paths diverged in the most critical of steps. The first one. In the Human Genome Project's approach, The genome was first divided into smaller, more manageable chunks, about 150,000 base pairs long, that overlapped each other a little bit on both ends. Each of these fragments of DNA was inserted inside a bacterial artificial chromosome, where they were cloned and fingerprinted. The fingerprints showed scientists where the fragments overlapped without knowing the actual sequence. Using the overlapping bits as a guide, the researchers marked each fragment's place in the genome to create a contiguous map, a process that took about 6 years. The cloned fragments were sequenced in labs around the world, following one of the project's 2 major principles, that collaboration on our shared heritage was open to all nations. In each case, the fragments were arbitrarily broken up into smaller overlapping pieces, about 1,000 base pairs long. Then, using a technology called the Sanger method, each piece was sequenced letter by letter. This rigorous map-based approach, called hierarchical shotgun sequencing, minimized the risk of misassembly, a huge hazard of sequencing genomes with many repetitive portions, like the human genome. The consortium's better safe than sorry approach contrasted starkly with Celera's strategy, called whole genome shotgun sequencing. It hinged on skipping the mapping phase entirely, a faster though foolhardy approach according to some. The entire genome was directly chopped up into a giant heap of small overlapping bits. Once these bits were sequenced via the Sanger method, Celera would take the formidable risk of reconstructing the genome using just the overlaps. But perhaps their decision wasn't such a gamble, because guess whose freshly completed map was available online for free? The Human Genome Consortium, in accordance with the project's second major principle, which held that all of the project's data would be shared publicly within 24 hours of collection. So in 1998, scientists around the world were furiously sequencing lines of genetic code using the tried and true yet laborious Sanger method. Finally, after 3 exhausting years of continuous sequencing and assembling, the verdict was in. In February 2001, both groups simultaneously published working drafts of more than 90% of the human genome, several years ahead of the consortium's schedule. The race ended in a tie. The Human Genome Project's practice of immediately sharing its data was an unusual one. It is more typical for scientists to closely guard their data until they are able to analyze it and publish their conclusions. Instead, the Human Genome Project accelerated the pace of research and created an international collaboration on an unprecedented scale. Since then, robust investment in both the public and private sector has led to the identification of many disease-related genes and remarkable advances in sequencing technology. Today, a person's genome can be sequenced in just a few days. However, reading the genome is only the first step. We're a long way away from understanding what most of our genes do and how they are controlled. Those are some of the challenges for the next generation of ambitious research initiatives.
2 important points of this movie. We know the genome of a human, and the cooperation of different scientists to construct the map of the human genome. This was the first example of cooperation. The second one was the discovery of new drugs against COVID-19. Cooperation from public and private companies. There was a call at the beginning of the pandemic between the scientists to send specific series of inhibitors of proteases, a specific enzyme of viruses. And all of these fragments were put together in artificial intelligence, and there were selected 10, 12 fragments, and one of them was the drug that you can find now in Turkey. So 2 examples of cooperation between the different scientists. Normally there is a big competition. Normally. I worked a lot during my experience in the muscarinic receptors, and at that time we tried to find antagonists for this type of receptor, and we were in competition with German scientists. And also I worked there for 2 years with the competitors. And it was a very, very strange history, yes. Okay. But why is so important in medicinal chemistry to know the genes map? Okay, with the small molecule, you know, it's not possible to fight this type of diseases. But we can prepare personalized drugs. If we know that a single patient has a problem with a specific enzymatic pathway, we can avoid the administration of specific drug to avoid side effects. Yes?
This gene stuff is also linked to the different outcomes of COVID in different patients. I mean,
yes, I understand.
learned that are the genes of innate immunity that cause the different symptoms. But it is also the link to treat the COVID.
Yeah, there is a specific group of scientists, and they are investigating why COVID can cause severe symptoms sometimes, and in other patients nothing, only a cold and just a little fever, and no inflammation of the lungs and so on. Perhaps it depends from our gene, yes, I think so, because you know from the gene, all proteins we see, the proteins are related to our gene, and we are different, and so perhaps we have a different target, different protein with a difference, for example, of only one amino acid, but it's enough because it changed the tertiary structure of the protein, can change the structure, and so perhaps this different point can respond in a different way to the virus. Yes. But from the gene, we can know, we can discover new target. And then we can try to identify new drug for these new targets. And also from gene derive the proteins, and also the proteins can represent a new target for the different diseases. In particular, medicinal chemistry is interested to find low molecular weight molecules, called small molecules. You know the new vaccine based on RNA technique. In my opinion, this is the most important discovery during the these last years, because to stabilize the messenger RNA is not so easy, because normally this molecule remains for a few seconds inside the cell. In 1 year, we had the vaccine, again, the COVID-19, and then really we can live normally. We can have our life normally. This is the most recent image of cell structure. There is a new technique called frozen microscopy. You know, the problem is the movement of the electrons of the elements, and so they frozen minus 80 degrees. I don't remember, but a lot. And you can see the membrane here, the double layer, the double phospholipids layer, the channels. It's wonderful, this image. Mitochondria here. Okay, you see on the book normally a representation, but not a real image. And it's really, for me, it's an extraordinary picture. If we know the structure of the proteins, we can discover very easily new drugs, because the problem is that we know the primary structure of a protein, but we don't know how the protein is arranged in the space. You know, the tertiary, the second, and the tertiary structure. In this way, you see the protein here. So perhaps really it's like a snapshot of the protein, and so you can design a compound able to interact with a specific target. Okay, today I have a lot of music, but I think it is interesting for you. Molecule is able to interact. this movie, you have seen how a molecule is able to interact with the target, and the forces, the bonds involved in this interaction. We'll see the different bonds, hydrogen bonds, pi-pi interaction, hydrophobic interaction. We'll see. It's a specific part of the introduction part. Here you can see an example. This is a... Hello.
Hello.
Oh, ciao. Buongiorno.
Beg your pardon.
Okay, thanks.
I've got a sample to be analyzed.
Grazie, Robby.
In the afternoon, pensi di andare in laboratorio da te in ufficio?
Sola, Mauriente.
Per quello.
Vuoi che ci vediamo?
Se vuoi, allora ci vediamo.
No,
perché risparmi a casa su. Allora niente, ha fatto bene andartelo qui.
Sì, ok, Robby.
No, tranquillo. Mi mandi il link per vedere.
Sì,
perché ho bisogno di capire che cosa devo fare.
Dunque, te cos'è che devo fare?
Io sono tutto didattico, però in Oriente avrei bisogno di fare qualcosa anche di quello.
Sì, va bene, ti mando allora. Ti mando
mandi il link, perché ora pensi di iniziare.
Mandami un WhatsApp adesso,
sì,
perché è finita la lezione e poi te lo mando subito.
Ok.
Adesso faccio una pausa e poi ti mando tutto.
Pensavo che l'avessi già fatta.
No, devo fare una pausa adesso tra un po'.
Va bene,
Okay.
Niente, allora aspetto.
Sì, dai, dammi un quarto d'ora, 20 minuti e arrivo.
Aspetta.
Ok, Robby, grazie. Okay, so we can see how an inhibitor or a specific enzymes have been designed. You see now. It's like a puzzle. You have to find the right piece, put in the right place, and if you are lucky. In this case, they synthesize an inhibitor, and this compound is used against leukemia. It's an anti-cancer compound because they need to inhibit, to have an inhibition of this pathway, and so they try to synthesize an inhibitor of this pathway. Okay. So, drug target. You have seen what is a target. It's a pocket, a specific point on a protein. But the problem is how we can find a small molecule able to interact selectively with this pocket. There are different ways. The first one is called automated screening. On the internet, you can find chemical libraries. There are companies specialized to have a collection of a million or a billion of chemical compounds. And if you have the the structure of the target on your PC with a specific software, if possible, to find virtually a molecule from this billion collection, 1, 2, 3, 4 molecules, virtually, and then you can order only these 5 compounds. And then you go in the lab, and then you try directly in the experiment if these 5 compounds are able to inhibit, for example, a specific pathway. This is the, you can have, if you are lucky, if you have money, also you have an automated screening. So it's a robot, you know, and during the night the robot works and it's able to try a lot of compounds, in this case not virtually. Automated screening. Screening is to make a choice, to try which compounds, between billions of compounds, is able to interact with a specific target. You can have the same plants, the algae. There are a lot of interesting compounds that we can use for the treatment of specific diseases. We can perform a rational design. We know the pocket. We try to fill the pocket with the chemical residues, able to establish particular bonds, hydrogen bond, hydrophobic bonds. We'll see what kind of bonds are important in the interaction with the target. This is a rational design, rational. I have the pocket on my screen, and I can design with a specific software a molecule able to interact. After that, I have to synthesize this compound, of course. And sometimes I can use the combinatorial chemistry approach. What is combinatorial chemistry? In one synthesis, I can get a lot of different molecules, not only one, but for example, the whole family with the different substituents on the aromatic rings, for example. Is a modern approach in order to have more molecules in one pot, in one synthesis. And finally, if you don't know the structure of the target, perhaps I know the chemical structure of an endogenous ligand. You know acetylcholine. You know acetylcholine is a very common neurotransmitter. And perhaps we can modify the structure of the acetylcholine in order to increase the potency of this compound if you want to have a more potent compound than acetylcholine.
But we can modify what?
Please?
We can modify what?
Oh, we can try structure-activity relationships. We can try to modify the oxygen atoms, the carbon chain, and so on, okay? We can introduce an aromatic range, for example. But we don't know the structure of the target, but we know the ligand. You know me, but you don't know how my wife is. Perhaps Tumiati wants his wife is a blonde, blue eyes, or perhaps not. But you know me. Oh, Tumiati is a very high, blue eyes, perhaps the same, the white. The approach is the same when you go in a black room. When you enter in a room, you try, oh, there is a window here, there is a window, there is a picture, okay, you have to, you try to know the conformation of the room, and you can make the same with the molecule. So you can modify the molecules in order to recognize specific point inside the target. So the problem are, the first problem, to find the target, the right target. The second problem is to find the molecule able to interact with the target. And when you have identified a molecule able to interact with the target, this molecule is called lead compound. A lead compound is the first molecule able to interact with a specific target. After that, you have to optimize your lead to increase the selectivity, for example, to increase the distribution inside the body, to avoid side effects, to modify the structure to avoid side effects. At the end of this operation, you have a candidate. So at the beginning, you have the lead compound. After the optimization, you get a candidate, drug candidate. Here you can see the long road to the new medicine. Normally 10 years. The vaccine was discovered in 1 year. You see the starting point is the design of a new molecule, but you have to know the target. The design, you see here the starting point. And then the synthesis, the screening, you have a cycle here, because you have to optimize also the lead compound. When you have the drug candidate, you have to evaluate the toxicity, preliminary toxicity. It's not enough to investigate your compound in a cell model, in a cellular model. It's not enough, because we need a complex system. It is necessary an optimal pharmaceutical formulation. This means that if you want to increase the absorption, the bioavailability of a specific drug, you need specific excipients. If you see on the box of a drug, you see the active ingredient, acetylsalicylic acid, for example, and then a lot of excipients. These excipients are important. You will see this topic in the next course of pharmaceuticals 1 and 2. These excipients are very important for the absorption and distribution of your drug inside the body. You have to perform new analysis methods if you want to investigate how much drug is on the blood. You have to perform a new analysis method. After that, you have the industrial development. You have to perform you have to optimize the synthesis. You have to obtain a lot of tons of your compounds. It's not enough 1 kilogram. You need tons of your compounds. And then phase 1, 30 patients. Phase 2, 300 patients. Phase 3, 3,000 patients. And in this time, you have to observe the side effects, the efficacy of your drug. At the end, registration to the health agency. You have to prepare a lot of data about the toxicity, the efficacy, the analysis methods, and so on of your compounds. All metabolites should be isolated and characterized. And the problem is, and the patent, you know what is a patent? A patent, we say brevetto in Italian. If I have a company and I want to develop a new drug, I need a lot of money. You see, preparation, synthesis, analysis, patients, investigations, and so on. And so I have to spend a lot of money in this road, but at the end I want to have a gain or not. But this long road is long, the time is about 10 years. And the patent covers normally 15 years. So you have only 5 years in which you can gain. You can have a gain about your drug. After that, there are the general drugs. Generico, we say in Italian. Generico is a general drug, is a old drug. The patent is expired. And so everyone can produce this drug. So long road, 10 years, the vaccine against COVID-19, 1 year. So you can understand the shortcut in the case of the COVID-19. Okay. Have you any questions? Comments? Okay, we can stop for 15 minutes, so we can take a cup of coffee. And then we start again about 11:30, okay, for 1 hour more or less. Okay. So. Here. Hello, dear students. We'll start for now. window. Mistake. Devo mandare una foto adesso. Caterina, ciao, chi era ieri? No, davvero? In italiano o in inglese? Grazie, grazie mille. Grazie.
No, ma sembra interessante, cioè difficile ma interessante. Sicuramente iniziamo a fare qualcosa che c'entra, cioè nel senso tutto c'entrava, però questa è proprio roba di farmacia, cioè patologia la fanno anche la mia amica di medicina, fisiologia la fanno anche a biologia.
I think all of them, but it's still one, but it's not great. And you do that. She takes time, you know, contributing to the race. Yeah, because I was like 24 February and the mom. How does it go? How does it go? Everything is fine, just boarding. Yes, I know. I want to sleep now. I'm not in the mood to start the new semester. Yeah. Yeah, nothing to do. Yeah. I'm very happy now. Oh, okay, here. Yeah. And you share an apartment? Yeah. How many roommates do you have? No. Flat sharing. Flatmate 3. And they're all schools. Are you planning to go back to India on the summer or something? I will go in September. That you did the service all right? Yeah. I just also now, because my visa was not anymore, it's just like 6 months, I think. It was 9 months, not 6 months. Yeah. Hopefully there will not be any more. Yeah. It's so much. They put it really also so far. They give the rules. But okay, 2 months I understand. Yeah, but like 4 now. 3, 4, 5. Or at least give us like, okay, 4 those within us. Exactly, but not like each time we go there is there is one. And Italy, it must not be that it's asking for the green card. Everything you do, you need to green card. You need to solve the green card. Too much. As a company, it's much more, like it's less severe if you want. They don't ask always for the green card. For everything. But if you want to answer to answer, any local you have to show them here. Yeah. One thing when I was in the vacations, and I was going out every day, I put my wallpaper as my green cards. Exactly, my God also is the same, but it's much more easier. Yeah. You go to center for some party. Now in India? Yes. Usually there's always more, I will show you one. Yes, and there is some place in which one. Some, yeah, from this okay, right. But it's like, to do is open. They want to before they close for public. Sometimes we realize it is something also and I do the labs also, so I cannot attend the lab like distance. Oh, really? Which one? Not one of chemistry, yes. So there's 2 courses mostly. The chemistry? And the but the Lawrence is easier than the Lawrence. And how is the test for it? For me, it was a little bit advanced because when I go to these sessions, I really didn't study so much. So it was like I just do it because of the exam. There is a time when you are fed up of but are you going to go on sometime? Yes, we go, let's say, almost like once, and it's fun to go on this. Smooth, right? It's much more better than bicycle. Yeah, it's more easy, more comfortable. Comfortable, you don't have to pedal until. Because as a bicycle, it's very annoying because there's a lot of people who walk, they're always stopped. It's fine. Because it's just you can drive on the road. Yeah, exactly. To get people must call not well, I do it from like the appointment from November until now it's not still not. I started in December. So you got it, yes? No? Okay. It takes you so much. Yeah, it takes
Okay. Oh. So we'll start again, and now okay. Prego, prego. Okay. So we start again. There is a question. Are we ready for DNA mapping? So we have seen the scientists have discovered the gene and the maps and so on. Okay. But the problem is, okay, you pay $50 on web, you send a sample of your cells to the company, and then you will receive your DNA mapping on a microchip. And then perhaps, oh, you have a problem with these enzymes, perhaps you can have a cancer, perhaps you can have a leukemia. The problem is, are we ready? You say no. Because you know, the people can react in a different manner. And so, in my opinion, I prefer to have not any news about my DNA mapping. Really, for example, in the past, I had cancer on a thyroid, yes, and my daughter had the same problem. So it's a genetic origin of the cancer. But I prefer to don't know anything about my gene.
You know, you can try to.
Yes, okay, you are prepared, okay, or you can ask for a solution, for a therapy, okay, maybe. But the people, we have seen the people during the pandemic. And so we have an increase in the use of anxiolytic drugs, really, antidepressants. And so, but the problem is another. You have your microchip. You go to try to find a job, and they ask you your microchip because they want to know eventually health problems related to your gene and so on. Your personal insurance, for example, you try to find, you want to buy a flat. You go to the bank, you ask money to the bank. Oh, please give me your microchip because I want to know if you have a long life or not. Yeah. Just to discuss, okay. But really, it's not, okay. Perhaps I want to know something about my future, okay. But the problem is all around. Banks, job, society. Really.
Now, is this procedure expensive? Because I know that it's thousands of euros.
50 euros or more or less.
Ah, really?
It's not so expensive. I don't know exactly, but you can see on the web, but 100 euro.
Yeah, 100 euros.
Yes, okay.
Once it was like 10,000.
I remember I knew too much.
No.
Prof, currently I've been searching for this method as well for DNA checking stuff like that. It was about $600 US for very specific DNA checking and
Okay.
like that. Yeah, $600. I think it is worth it if you're really related, if you're really worried about your
Okay.
Yeah.
I prefer to go on holidays 1 week.
Exactly, me too. I have nothing to know about my health. Yeah, I prefer that one too.
It's my opinion.
Yeah, me too.
Okay. And this is a very old drug against infection in the urinary tract. But it was the first drug that reported on the sheet the problem related to the specific genetic characteristics. You see, the glucose 6-phosphate dehydrogenase deficiency. And this problem is related to a certain ethnic groups. And so it was the first personalized drug. No, la nitrofurantoina for the infection of the urinary tract. It's a very, very old drug, but it was the first that reported this type of relation with genetic characteristics. Yes. You see here predominantly Negro and more rarely Caucasian population nitrofurantoins, like many other therapeutic agents, may result in hemolysis, the appearance of so on and so on. Okay, so it was one of the first that reported the problem related to the genetic characteristic. So now we have some definitions. What is a drug? A drug is a substance from chemical synthesis or natural sources that can be used to prevent, to alleviate, and treat diseases in humans and animals. What is a diagnostic? A compound used to identify diseases quick swabs, for example, or a stick to measure the sugar in the blood. Diagnostics are very important, you know. So, drug is the active ingredient. Medicine is all formulation, active ingredients, more together with the excipients. So medicine is a medicinal preparation, finished medicine. We can define a drug through three particular characteristics: chemical structure, pharmacological effect, and type of diseases. Chemical structure, we'll see during the course what is a penicillin, what is a steroid, what is a peptide, and so on. Pharmacological effects, a diuretic, for example, an anti-inflammatory, and this type of definition is typical of the pharmacology course. This is an hormone, you see the chemical structure, very similar to cholesterol, you see. Penicillins and cephalosporins, we'll see in the course the chemical structure of these compounds, of these families. Pharmacological action, hypnotics, diuretics, vasodilatators, and so on. Physiological classification based on action site. Drugs for CNS. Why the drugs for CNS represents a particular class? Because the drug should be able to reach the neurons inside CNS. We'll see that there is a barrier able to divide the brain from the body. Blood-brain barrier is called BBB. And only a few number of drugs is able to cross the BBB, blood-brain barrier. Okay, you can say pharmacodynamic drugs, drugs that interfere with the body's function, vasodilators, anti-allergics, and so on. Chemotherapeutic drugs, it's a special part of our course, antibiotics, antifungals, anti-cancer. We try to hit selectively the external agents, for example, bacteria, viruses, but we can consider the cancer cell as external agents, but you know that a cancer cell is an our own cell, and so it's not so easy to have a selectivity to hit the cancer cells because they are very similar to our health cells. Okay, and then miscellaneous drugs, you see. Other. What happens when we take a pill? I have just a little bit fever. I decide to take an aspirin. I take the pill, tablet. The first step is the absorption. The tablet reaches the stomach or the intestines, and then the drug should be released from the matrix, cellulose, excipients, different excipients. And the free drug should be able to cross the membranes of the cells present in stomach, intestine, in order to reach the blood flow. It's not an easy phase, because sometimes perhaps you have eat a lot and your stomach is full. The drug can interact with the foods, different foods. We'll see that the absorption phase is regulated by particular transport systems located in the intestine cells. But you need about 1 hour to have the effect, you know. And when the drug is present in the blood flow, the drug can be present in 2 different forms, as free drug or bond drug to the protein, to the blood proteins. You know that in the blood there are a lot of proteins, albumins, red platelets, and so on. And then normally, 90% of drug is bound to the blood proteins. You see in this expression, you see drug plus proteins, you get drug bound to proteins, DP. Normally, 90% of drug is bound to proteins, and only 10% is free. But only the free drug is able to cross the different membranes to reach the action site, to reach the secondary action sites, and so the side effects. Only the free drugs can be excreted, eliminated. Only the free drugs can be inactivated. And the bound drug is like a reserve, because you see in the expression is an equilibrium. And so if the free drug 10% decreases, you have the reserve. Is important to restore the right concentration of the free drug inside the blood. A lot of new drugs, of new molecules, of new candidates were eliminated because they presented a too strong bound with the proteins. For example, 98% is bound, only the 2% is free, and the 2% is not enough to reach the active concentration in the blood. You have studied microbiology, and you know the minimal active concentration against a bacteria, and we have to reach this value if we want to fight, to kill the bacteria. If your antibiotics, for example, is bound too strong to the blood proteins, it's not possible to have the right concentration of the free antibiotic in the blood. The free drug can also reach the depot sites. Depot sites. Depot sites normally is represented by fat. Fat are located under our skin, and these sites represent another reservoir. If you want to have a long active drug, for example, there are drugs that you can take only one time in a week, and you have a very long activity. This administration is able to cover 1 week of therapy, because part of the drug can remain in the fat and slowly released in the blood. Depot sites represent also a problem in the doping investigation. If an athlete uses hormones, growth hormones, steroids, to increase the muscle mass, these hormones normally stay in the fat for 1 month or more, and so you can find traces of these hormones in the urine after 2 months. Because now we are very sensitive equipment, very sensitive, and we are able to find this type of doping. The most important point is the blood level of the drug. You have to reach the active concentration of the drug and the level of the drug depends on 4 different parameters. Okay, the first one is the dosage, It's important to take the drug in the right dosage. It depends on your weight normally. The second point is the absorption. The drug should be absorbed to reach the blood flow. Distribution. The drug is distributed in all the body by proteins. Metabolism. A drug is considered an exogenous compound, and so our body tries to eliminate as soon as possible this type of this new compound, this external, this host compound. It is an enemy of our body. And so our metabolism is able to transform the chemical structure of a compound, every type of compounds, drugs, pollutants, in order to maximize the elimination of these toxic compounds, of these exogenous compounds. And so our metabolism is able to increase the hydrophilicity of a host compound, of an exogenous compound. How? By introducing, for example, OH groups, alcoholic groups. But why we need to increase the hydrophilicity? Because we want to eliminate this compound by urine. You know, urine is composed by water, 90%. And so it is necessary that our compound, it is necessary its solubilization in the urine, the water. But we'll see the reaction, the metabolic reactions. And the last phase is the elimination by urine, by lung. And so we'll see. You can see this acronym, okay, here, acronym ADME. A for absorption, D for distribution, M for metabolism, E for elimination. This acronym describes the characteristics of your drug. And normally there is another letter T, ADME, for toxicity. Absorption by injection, tablet, you see. Distribution, okay. Sorry, this slide is in Italian, but you can understand plasma is blood. Legato is bound, libero is free. We have the equilibrium between these two forms. Distribution in all the body. Metabolism and elimination. Liver, kidneys. But at the beginning, when we have a new drug, and we want to investigate if this drug is active or not, can we have some information about ADME in advance? It's possible or not? There is a special software. You can find this software at this web page. And if you put your chemical formula of your compound in this software, you can have some information in advance about intestinal absorption, solubility in aqueous solvents, ability to cross the blood-brain barrier, the percentage of drug bound to proteins, hepatotoxicity, and so on. But why is it so important to have this type of information in advance? Because a company, when started to investigate a new compound, wants to know if it's worth or not to spend a lot of money for the development of a new drug. If you have a new drug, okay, in the cells experiments, the new drugs is very active. Okay. Oh. In attesa, just a moment. Abbiamo anche un ospite. We have a guest. Okay. Okay. They start with an investigation of a new drug, okay. And we arrive in the phase 1 with about 30, 40 patients, okay. We use these new drugs, and unfortunately, this drug is not absorbed in the intestine. It's not able to reach the blood flow, or otherwise is too strong bound to the blood proteins. So the company can lose a lot of money. And so it is very important to have this type of information in advance. There are other rules that allow us to know if a compound can be active or not. Lipinski rules. These scientists investigated all drugs in the pharmacopoeia, more than 2,000 drugs. During the night, perhaps. he didn't sleep, and so checked the pharmacopoeia, and he elaborated some rules in order to have an optimal drug. How to have an optimal drug. Molecular weight less than 500. If the molecular weight is too high, there is a strong difficulty to cross the membrane. No more than 5 hydrogen bond donor groups. No more than 10 hydrogen bond acceptor groups. A calculated log P. P is a coefficient related to the lipophilicity of a compound. The ability to solve this compound in organic solvents, chloroform, ether, and so on. We'll see this particular coefficient because it's important to have an high p-value, high lipophilicity to cross the membranes. But why? Because you know the membranes are formed by phospholipids. Phospholipids are a long carbon chain, if you know. And so we need a similar behavior of our drug in order to cross these double layers. The vast majority of drugs registered in the World Drug Index, WDI, fall within this criteria. Violation of at least 2 rules results in a high risk of non-developability. Okay, and then some recommendation, but just to know, oh, this is important. There are another scientist, Weber, in 2002, investigate the importance of flexibility of a molecule in order to reach the target. You have the example in this picture, a snake. This snake is able to reach its hole only in a line form, you see. But when the snake is inside its hole, it can change the form. It's like a drug. The drug is able to have the line form to cross the membranes and then to change conformation when interact with the target. So the conformational freedom is very important. needs to be flexible to reach its site of action. And so here you can find a lot of parameters about this characteristic, this flexibility. These rules suggest that it may be advantageous to make the structures rigid to reduce the number of rubble bonds. Okay, bonds. And then a lot of values. These data are not important for us, but it's just to give you an idea about the complexity of the problem in the absorption phase and in the phase when your drug try to reach the target. You see Lipinski rules, Weber rules for CNS targets, and so on. Okay, here we can see some example. Tegretol is an anticonvulsant. Diazepam is an anxiolytic, is a benzodiazepine. You have an anti-cancer, an antiviral. You can see here the molecular weight. You can see here the hydrogen bond donors, the number of the hydrogen bond donors for each of these molecules. Hydrogen bond acceptors, log P, partition coefficient, the lipophilicity of each compound. And so, in this way, you can have a complete picture of your molecule in order to have information about its behavior inside the body. Okay. Have you any questions at this point? It's clear for you? Have you any question at home? I can see here. Have you any question? All clear so far? Okay. Ciao Jude. Ciao Stephanie. Okay. Okay. So I think 20 minutes and then stop. Okay. Because these are all new concepts and I think it's enough. Okay. The process of drug action. The first step is the application. If you have a tablet, you have to take the tablet. If you have an injection, you have to make the injection. So, in this phase, the drug is introduced into the body through an appropriate route and an appropriate form. It's impossible to take a tablet by injection, you know. But sometimes happens. Yeah, really. I try to take this tablet by injection. How? Oh, I make a pulver and then I try to solvidize. Then no, it's not the right way because the excipients are quite different. So we have to take the tablets orally and not by injection. First step. Second step, pharmaceutical phase. The active ingredient is contained in a matrix together with the excipients. It is necessary the release of your drug from the matrix. in order to have the drug available for absorption, we need free drug. Then we have the pharmacokinetic phase. This means that the drug is already absorbed, is present in the blood, and is transported in the whole body. If it is able, also in the CNS, also in the brain. In this way, your drug is able to reach the active site, the target. When your drug is present in the front of the target, is very close to the target, you have the pharmacodynamic phase. Your drug is able to interact with the target to give the therapeutic effect. So we have seen the different steps from the application to the therapeutic effect at the end. In the middle, there are three different phases: pharmaceutical phase, pharmacokinetic phase, distribution, pharmacodynamic phase, interaction, target interaction. So, once again, pharmaceutical form, dissolution, drug in solution, absorption, distribution, metabolism, excretion, drug in the body fluid, drug receptor complex, and finally, pharmacological effect. The slowest stage determines the speed of the entire process. If the solubilization is too long, this delay can influence the whole process. For example, another important concept is reported in the slide. Bioavailability is the most important concept. Bioavailability defines the percentage, the fraction of the administered doses of drug that reaches the circulation, the blood flow in an active form, and is therefore available for systemic action. Bioavailability is the most important concept. It describes all these steps: dissolution, absorption, blood concentration. So when you hear this word implies all these steps. It's the most important word for us. Once again, bioavailability represents the amount of drug available to the body after the administration of drug through different ways, okay? You can see here, orally, by mouth, Orally, we say also per os in Italian, but per os is Latin. Os oris is a mouth, is a Latin word. And normally in Italy, doctor, when he writes per os, is by mouth, orally. If you inject a drug, you have 100% of bioavailability because you reach directly the blood flow. Normally, orally, you have only about 40%, 50% of bioavailability. This means that you need a higher dose of drug orally in comparison to the injection for dosage. If you use, for example, an antibiotic, we need 1 gram per os orally, and by injection perhaps the half. Because by injection you have 100% of bioavailability. Yes?
So a lot of antibiotics, like for pneumonia, are administered intravenously because less drug, less side effects.
Yes, and sometimes some antibiotics are not stable at acidic pH in the stomach, because you have to remember, and we'll see that in the stomach you have the 1.5 pH value, and a lot of molecules are not stable at that pH value, and so it is necessary the injection directly on the blood because the pH is 7.4. This is another important point, and we'll see later. Some penicillin are stable only by injection, but you know it's not so comfortable to the administration of a drug by injection. I remember when I was a child, my father with a syringe followed me inside the house because in that time I used antibiotics by injection, so really.
I did for pneumonia.
What happens in the blood flow? We can see in this plot, you have the time and then plasma concentrations. At the beginning, you have a low, when, for example, you take a tablet of aspirin, you have a low concentration of acetyl salicylic acid, but this concentration increased, you see, is able to increase, and to reach the peak after, for example, 1 hour, 2 hours, and then again the decrease of the curve. Because you have a loss of drug due by metabolization, elimination, and so your drug can remain for a limited time inside the blood. But it is important that the peak of the concentration is over the minimal effective concentration, minimal active concentration. We have to reach and to be over. We have to be over this value. But we have not to reach the toxic concentration. Sometimes the excipients, in this phase, the excipients are very important to regulate the release of the drug inside the plasma. So excipients can influence the release of the free drug in the blood, but also in this phase are important the physical and the chemical characteristic of the drug, solubility, lipophilicity, and so on. So here we have the entire picture of the drug administration, of the drug assumption. At the beginning, drug dosage, it's important, the right quantity, absorption, and then the concentration of free drug in the extracellular fluid, okay, blood flow, serum proteins binding, bound drug to the blood proteins, binding and deposition in tissues, depot sites, biotransformation, metabolization, metabolites, active metabolites, inactive metabolites, excretion, mainly by kidneys, drug concentration at the active site, receptor occupancy, and finally pharmacological effect. This is the entire picture of all how we can get the final effect. It's not so easy. If we have some problems during this long phase, perhaps we don't have the pharmacological effect. Okay, this is the last slide for today. Yes. Routes of administration. We start from the beginning. The first phase, you see the absorption. We have different ways of administration. Enteral occurs via gastrointestinal system. You know the tablet, the most common, but we have also sublingual, under our tongue. It's possible to put a tablet under the tongue, and then you have an absorption directly in the blood flow without the passage in the stomach. And so you have a very fast effect. If you have an headache, sometimes you can take a painkiller, then you put the tablet directly under the tongue and you have an effect in 10 minutes. In this way, we can use also some drug for the heart failure, vasodilatators, very potent vasodilatators. Nitroglycerin is an explosive, but also explosive is also a drug, and is a very potent vasodilator. We use this drug in the case of infarct, heart infarct, because we need a vasodilator in this case very fast. So we can use this drug in the case of infarct or angina pectoris is a pain due to the loss of oxygen to the heart muscle. We can assume our drug by nose, rectal, we can have a parenteral injection, 100% of bioavailability, not first pass effect. This means that if we take a drug by injection, we have not a passage through the liver. You know that every compound assumed by orally, it's absorbed in the stomach and then reach the liver. In the liver, we have a lot of enzymes able to change the chemical structure of our compound. So if we want to have a very high bioavailability, we have to avoid the passage, the first pass effect. You see subcutaneous injection, intramuscular, intraperitoneal, intravenous, intraarterial, intracerebroventricular is inside the CNS. We have to cross the bone and then put directly to the drug inside the CNS. Okay, it's not so comfortable. Okay. In this case, we have to use a drug which is not able to cross the blood-brain barrier, and so we have to inject directly this drug in the brain. Miscellaneous routes, aerosol, topical, transdermal, implant, and so on. An important concept is the administration is critical for several pharmacokinetic, toxicological, pharmacodynamic parameters. Okay. So, have you any questions for today? Any question at home? Ramona, no? Okay. Okay, Zara? Okay, Stephanie? Okay. Okay, so we'll see next time. Thursday in the morning, about 10 o'clock, okay? We'll be here again, I don't know, in this classroom, perhaps or in the other one, 1A or 1B, I don't know, okay. However, we'll see. Okay. So, have you a nice afternoon? I am involved today with Caterina in Alma Oriental, a lot to do. Okay.
Today is the open day.
Yeah. No, yes, virtual open day, you know, for the high school students. Oh, a lot of questions. Okay, so have you a nice afternoon. See you next time. Bye-bye. Bye-bye. Okay, stop the registration. Interrompi.